you know, and we will learn more about this correction factor ahead. But what it is, you know, this shows decreasing. So this is a residual product of a graph of a function after matrix algebra treatment. So I found that myself. And what it's showing you that the genetic fitness of this graph is a little bit higher than this graph over here, okay? And since it is a correction factor, you are subtracting this amount and bringing the consistency in your analysis from one segment to another segment. You have the stability in your results, you know. So that is the find. That is the find for this. Sure, thank you. You know, that is the find in this experiment, because if I don't apply, and I'm going to show without applying the correction factor and applying the correction factor, what is the difference, okay? So that brings us to the next one, reverse engineering. This person, Dr. Mendoza, you know, he has published extensively, you know. I mean, you know, and I was talking about visual. I took the difference of frequency between dominant and recessive and divided that by 12, okay? And that gave me a value of this. So the difference between, you know, so the average was this, and the difference between dominant and recessive was 88, okay? I know it's very easy, but when I went there, when the data was in front of me, I needed a prayer, you know, because only because of those two concepts of mathematical genetics, I was able to combine the difference between frequencies and then take an average of that. Okay, so equilibrium, you know, when I started my after MBA, you know, I needed a direction, and this abstract is very close to my heart, you know, because it taught me that there are 2, 1 plus 1 minus 2. So I can say that in LWR, the genes that are responsible, or the DNA sequences that are responsible for, what you call is 300K tumor size, you know, are kind of similar to other larger type of tumors, you know, but we see that it completely misses the smaller tumors, like, you know, 25 and 75 and 100K tumors. It doesn't come close at all, you know. And I have also tried to summarize that there are two different populations, and you know, so because of different populations, you know, maximum likelihood says that since 300 to 600 is so slow, the gene activity is so slow. If you take any of them to predict, you know, what's in the fast action, you cannot. This one doesn't it doesn't miss as much, but what it is that it does come close as far as, you know. So you can take a 300 micrometer tumor size of BC and plug that up into equation, the frequency observed for that tumor, plug that up into your equation, and it gives you a frequency pretty close, you know. So you are also getting an idea what is the rate the cell division is going on, you know. So that's what you're trying to figure. If you develop a medicine, you need to know at what rate cells are dividing, cancer cells are dividing, so you can try to stop, you know. And this mathematical equation tells you at what rate they are dividing, you know. And I have made some of the conclusion there. Okay. Now, this is the complete thing, you know. This is on the first page of her thesis that I found. And it kind of touched me, you know, because I used to think that why people do PhD, you know, 7 years of torture. And right away she wrote, the reason she's, well, I took her words and I translated into action. And this is the action, okay? So this is meta-modeling, okay? This is the original data, okay? This is the original data. It's above threshold, okay? So that's number one we see. Here it shows that both of them are above threshold. That is not true. That's an error, actually, you know. this one is above threshold. And actually it shows that there was a gap in between, which as the fitness increases, the gap between the dominant and recessive, semi-dominant and recessive decreases. But this one doesn't say that. It says that actually the difference stays right there. That's a big difference, So you have a bunch of sentences, and you need to put it in action, okay, and make it a very clear, crisp interpretation, okay. So this one helps you do that, meta-modeling. And what we see that none of these guys go like this. It doesn't go like that. It doesn't go like that. It keeps increasing, right? So since we didn't get any plateau, there may be a structure similarity because I'm using this to predict this guy. I'm using this to predict here. I'm able to predict, but the T30, each of these cell types, I'm dividing LWR BC divided by LWR. At one time, what I saw that one of the frequency of LWR for a certain cell size is 8, and the ratio there was between mean and the median. Okay, another frequency for a cell type, cancer cell type, was 11, and the ratio there was between these two. So sometimes it helps not to just look, mean, median, skewness, and kurtosis, but it's important to see what number is close to what number, That is important, because if I didn't have my eyes like that, I won't be here, And I just saw right away, oh gosh, the ratio which I have gotten by this number of LWR is between these two, and this number is between these two, okay? So there is something about 8 and there is something about 11 that I need to consider, And look at these numbers. Look at these numbers, And if I go to the next one, these are the same numbers, Even though I've taken the BC frequency divided by 11, and this by 11, and then this by 8, and this by 8. All these numbers are same. Why is that? Because the minimum is this and maximum is 3.0. So it doesn't have too much variability to go something. So there is no biological effect here. It is the random event due to the small, variability. That's what my interpretation is. Okay. Now, if I go to the next one, look at this thing. I added 11 and then took a ratio of that, and suddenly I have the mode. This is the first time I found mode, and then, I did 10 simulations. Remember the theory, median of a ratio is Gaussian-like, So I took that theory and I'm applying here. And what I see that I did 10 of those ratios like this. I took a median on those medians, and suddenly what I have is a mode, and that was a median value previously. So I'm watching how my data is changing closely, and when I'm getting a median, when I'm getting a mode and not, And the reason is happening because of competition, Cells compete among each other to be there, So So by doing this way, I figured it out that what numbers I need to work with and what numbers I did not work with. And this is the way I did my data mining, looking at the proper numbers to figure it where it fits, Where is the another one? Oh, okay. Right. So we have seen this before, and it's a pretty picture. Well, nice. Okay. So now what I do, that now I'm going towards the second hypothesis, compensatory response. And what it is that in the first shot, I know